Thank you so much, Squire, for such a very kind introduction. Thanks to the ASBMB for this award. This was a really surprise to me, and I'm really honored and humbled by receiving this award. So what I thought I'd do is sort of give you a bit of a view of some of the work that we've done in the lab. But before I do that, I wanted to acknowledge and take a moment to acknowledge Marion Sewer, who was recently passed away, and she was a faculty member here at UCSD, not too far from here. She was taken too early, and she represented one of our leading stars in the field. I also wanted to remind you that on this date, Martin Luther King delivered his final address the night before he actually was killed. And this is referred to his mountaintop speech. And you can see there what he said about seeing the promised land but maybe not getting there. I also wanted to acknowledge and really laud Ruth Kirschstein, for whom this award is actually named. She was the first female director of the NIGMS. She was really well known for her work in vaccine safety, but also a tireless advocate for training of women and minority scientists. And she actually published in 2001 this call to action. This is a viewpoint in Public Health Report. And she pointed out the health disparities and really pushed for more research in health disparities. And I'm really humbled by being in the same podium with these former Ruth Kirschstein Diversity Award winners, all pioneers and leaders in the field of diversifying science. And so I'm really pleased that the ASBMB has chosen me as the award winner. So I thought I'd take a little bit of time and tell you about one project that we work on in the lab, trying to understand how mast cells get activated when they get triggered by allergen. And so the work in my lab has been done not by me, but by a number of students and postdocs. It's just a word cloud of all of my co-authors over the years. And most of my work actually revolves around trying to understand this particular kinase, tyrosine kinase called ITK, and how it regulates immune function, and in particular, how it regulates effector function in T cells. But I thought I'd spend some time talking about something else today. And so if you actually do a word cloud on my publications, you can see kinase is very prominently represented. So the word that I'm talking about today is really about allergies. And we can think about allergies if you think about your the colleague next to you. You'll be sure to find out that almost 77% of all Americans are affected by allergies or asthma. 50% who have asthma in the household, and 29% who know someone in the disease. So the person next to you, if you're sitting right here, most likely knows someone or has allergies or asthma. So it's a huge public health problem. Particularly affects people who live in urban areas. And so the cell types that are involved in this disease have been really fairly well described. And they include an intersection between a number of immune cells that produce cytokines when they're activated and cells that respond by producing pharmacological agents when they're activated. In particular, the mast cell here is a critical player in this particular response. And so a number of investigators and companies have tried to explore what is it about the mast cell that we can manipulate that would allow us to block an allergic response. And so a number of agents have been developed over the years, including starting on your right, corticosteroids and beta-adrenergic agonists that really block the inflammation that is caused by the release of these pharmacological agents. And then more recently, anti-leukotrienes that block the effects of leukotrienes that are produced by cells like mast cells and eosinophils. And then proteins such as omalizumab, which targets the antibody that actually triggers the mast cell. And the antibody is IgE, or immunoglobulin E. And IgE binds to a receptor on mast cells called the Fc receptor type 1 or high affinity receptor. And when that IgE binds to the mast cell, it interacts with allergen, and that allergen then triggers the mast cell to be activated and releases pharmacological agents. And so this story starts with the combination of a number of students who participated in this work, and I'll try to point out their names as we go along. We also collaborated with a chemist, Blake Peterson, and a neurobiologist, Tomo Shirao at Gunma University in Japan. So it starts with a interaction I had at a meeting very similar to this with a friend of mine, Jim Trevine, who was at Abbott Labs. And he pointed out that he had a compound, which he called BTP, that was able to inhibit inflammation. And this is the structure of the compound. And so he said, if you guys are interested, I can give you some of the compound. You can try to get a better understanding of how it works. And so we were very interested in sort of the signaling mechanisms that affect immune activation. And so we decided to spend some time looking at this compound. And so what we and others have found is that this compound, BTP, can actually inhibit the allergic response. It can inhibit inflammation of inflammatory cells into the lungs, for example, during allergic asthma. And so we wanted to find out whether it also directly affects the mast cell. And so we did, and when I say we, I mean Kit Law, did a simple experiment where he took IgE and injected it into a mouse. And then 24 hours later, he gave that mouse either the compound or vehicle, and then challenged that mouse with the allergen, which is recognized by that IgE. And what usually happens is that IgE then triggers an allergic response from the mast cells, and you can then measure the amount of histamine that's released by these cells. And what you can see here is that when we treat these mice with BTP, we can significantly reduce the amount of histamine that's released by mast cell activation. So mast cells can produce histamine, but they also produce other pharmacological agents that result in a reduction in temperature during an immune response in terms of allergies. And so we did another experiment where we asked whether the compound could actually affect passive systemic anaphylaxis. This is what happens when allergen gets inside the system intravenously and causes massive anaphylaxis and shock. And you can see here that under normal circumstances, animals that have been triggered rapidly lose body temperature over time, and that can be significantly blunted by treatment with BTP. So this compound is able to significantly affect both mast cell release of histamine, but also the resultant temperature loss that you have during a systemic anaphylaxis response. So we could conclude then that BTP is able to inhibit mast cell function in vivo. And so we wanted to find out whether it also inhibited other things. And so in data I'm not able to show you because of time, we can show that it inhibited calcium influx in cells, degranulation of these mast cells, and cytokine secretion. And so all of that is all of those experiments were done so that we could try to identify what is it that this compound was actually doing to the mast cell in order to affect its activity. And so one of the things we wanted to do is identify the target of this particular compound. And we wanted to try to do this using biochemical approaches. That is, what proteins bind directly to this particular compound? Well, we first needed to find out which portion of this compound is most important for its activity. And so this is where we got to the point where we decided, well, we weren't chemists, and so we needed to collaborate with somebody who was able to actually look at this compound and make some derivatives. So before we get to that point, I sort of wanted to sort of just let your mind take in what I just told you about the effect of this compound and remind you, or tell you if you didn't know, that Marie Maynard Daly was the first African-American woman to earn a PhD in chemistry in the US, and she got this at Columbia in 1947. And she was a faculty member for a long time in New York. So coming back to this problem that we had, which is what is the target of BTP? And so we collaborated with Blake Peterson and his student Laurie Mottram, and he synthesized for us a number of derivatives of BTP that would allow us to probe different functional moieties of the compound to see which portion of the compound is most important for its activity. And so we looked at the trifluoromethyl groups at the 5 and 3 position of the molecule, and what we found is that this portion of the molecule was not important for its activity because we could change that and still have the same activity as the parent compound. But if we change these, any one of these two trifluoromethyl groups here, we get differential effects. So if we remove both the trifluoromethyl groups, we completely lose activity. Or if we remove the 3 position, we lose activity. Or if we change that to a methyl group, we lose activity. But if we maintain the trifluoromethyl group at the 3 position, then we maintain its activity. And so this is just summarizing a number of studies showing the structure-activity relationships of this compound. We use that information then to modify the compound by adding it to a linker that allows us to be able to purify proteins that would bind to that compound. But before I tell you about that, I just wanted to tell you another piece of information about 2 very important pioneers in science. These are all women. And the reason I'm just highlighting women is because last month, March, was Women in Science month. I don't know why it has to be a month, but it's Women in Science Month, so I thought I'd highlight some women in science. This is Lydia Villa-Komarov. She's the 3rd Mexican-American woman to receive a doctorate in molecular biology and genetics, actually in science, at MIT in 1975. And Shirley Ann Jackson, who was the first African-American woman to earn a PhD from MIT in nuclear physics in 1973. So coming back to the question at hand, we showed that the trifluoromethyl group at the 3 position on BTP is very important for its activity. So that allows us to now be able to modify this compound and find out what it binds to. And so that took us down a road where we were in a complete black box. We didn't know what we would find. But Jason Mercer, who was a graduate student in the lab at the time, took this challenge and grew up large numbers of cells and devised this experiment where we could now couple this compound to a bead using a biotin group here in streptavidin beads, or a control linker, and take proteins that we extracted from cells and pass those proteins over these beads and then purify proteins that bound only to the BTP compound. And that's when ISHT, as they say, got real, because now we could identify a particular protein that we could then determine how it worked in mast cell activation. And that protein is called drin. So drin stands for developmentally regulated actin binding protein. It's been well described in the neuronal system because it interacts with actin and is involved in the formation of dendritic spikes, but had not been described in terms of the function outside of the neuronal system. And so what we found, I'm summarizing a number of studies here, we found that drin actually bound to BTP directly through this central actin binding region here, because if we use proteins that did not have or had truncated actin binding regions, we lost binding activity. We also found by some very sophisticated sequence gazing that BTP actually interacted directly with drin via these 2 licenses here, because if we mutated these 2 licenses to methionine, we completely lost binding of BTP to drin. And so we could then determine then that this compound, BTP, interacts with drin in this actin binding region, specifically with these 2 lysines within that region. But the question is, how does actually BTP affect drin function? And unfortunately, we don't have a good idea as to what drin does. It doesn't have any enzymatic activity. What we do know is if you overexpress drin in cells, you end up with massively reorganized actin cytoskeleton. So we decided to take advantage of some microscopic studies and to try to investigate whether this activity of drin was affected. So this is what happens if you overexpress just GFP alone in adherent cells. And I don't know if you can see that there very carefully, but you can see that actin filaments are formed, and really the cells have a nicely well-formed actin filament. If you overexpress drin, this is what you see. You see massive reorganization of the actin cytoskeleton, and drin is actually tagged with GFP, and you can see that it completely co-localizes with F-actin. If you treat these cells with BTP, you significantly reduce this reorganization of F-actin in terms of these filopodia-like extensions. We actually could show that these filopodia-like extensions are actually reduced by BTP treatment. And the mutant that we identified that could no longer bind to BTP was not affected by drin. So that mutant is shown over here. You can see you still get these actin reorganization, and that is not affected by treatment with BTP, whereas the control mutant is affected and the wild type is affected. So in a number of different studies, we could show that knockdown of drin in cells reduces calcium signaling in these cells. And this is using shRNA. And so what we wanted to know then is, how does this affect mast cell function? But before I do that, again, I wanted to take a brief interlude to remind you of two other very prominent female scientists. The first African American and first Hispanic woman in space, Mae Jemison in 1992, and Ellen Ochoa in 1993. The 90s was a very good year for women in space. And they were pioneers in NASA in getting on a space shuttle. So what I've shown you so far is that BTP inhibits mast cell function, inhibits degranulation and cytokine secretion. This is data I didn't have time to show you, but also inhibits calcium signaling. And so we wanted to find out whether this compound was able to affect mast cells by affecting drin. And so we used the information that I told you to identify the moieties that are important for BTP function. We purified the drin protein. We showed that it could bind to these two lysines. And then we generated transgenic mice, or actually knockout mice, inbred strains of mice, to try to understand drin function in vivo in mast cells. And so we generated mice that lacked drin and did the same experiment that I showed you before. Instead of now treating these mice with the compound, we're just treating mice that either have drin or are missing drin. And so again, the experiment is the same. You can inject IgE into these animals and then challenge them with the allergen, and you can see whether histamine release is affected. And what you can see here is a mirror image of what we saw, or actually the same as what we saw for mice that are treated with BTP. That is, there's a reduction, significant reduction in the amount of histamine that's released by these mice in the serum after challenge, allergic challenge. If we examine these mice using a passive systemic anaphylaxis model, again, this is what I showed you before. These are mice that are control mice, and the drin deficient mice don't really lose body temperature, and that overlaps significantly with the mice that are treated with BTP. So what we could find then is that in the absence of BTP, we get reduced mast cell function, we get reduced calcium signaling, and we also get reduced allergic responses in vivo. So these experiments suggest then that drin is really important in mast cells for regulating calcium signaling, degranulation, and cytokine secretion, and that BTP is able to affect mast cell function by actually interacting with drin. So in the last interlude, I wanted to remind you of two other women who were the first Hispanic and the first African American female surgeons general in the US, that is Antonia Novella and Joycelyn Evers, again in the 1990s as head of the HHS and the Surgeon General of the US. So one of the things I told you about BTP is that it affects drin function in order to affect actin reorganization. And so we wanted to find out whether the drin deficient mast cells had a similar defect. And so we looked at mast cells that we generated in vitro and examined the amount of filamentous actin in these cells. And so on your left, you can see how we measured the amount of F-actin by flow cytometry, and you can see that wild-type cells actually had less F-actin than drin-deficient mast cells. And if we look at them on a confocal microscopy, this is a low-resolution image, this is a higher-resolution image, you can see very immediately that the wild-type mast cells have F-actin around the perimeter of the cell, whereas the drin-deficient mast cells have more F-actin, and it's actually more distributed throughout the cell. We actually quantified this using an algorithm, and you can see here that in wild-type mast cells, most of the F-actin is at the periphery of the cell, but if we look at the drin-deficient mast cells, you can see that most of the F-actin is actually distributed throughout the cell. So there's a major difference in how the cell is managing to reorganize its F-actin in the absence of drin. When you stimulate wild-type mast cells, you actually get a reorganization of F-actin. And you can see here, if you quantify those cells over time, that F-actin is reorganized and is moved towards the middle of the cell and then recedes back to the perimeter of the cell once the cells have been activated. However, if you look at the cells that lack drin, you can see that F-actin is also reorganized, but it really doesn't change and return back to the perimeter of the cell. It actually end up having more F-actin throughout the cell. And we think that's really important for affecting mast cell degranulation and function, because without being able to reorganize and disrupt this F-actin superstructure, the cells can't release their contents and therefore degranulate and lead to allergic responses. You can actually quantify these differences by subtracting these two plots here, and you can see that throughout time, there's a significant difference between the drin-deficient mast cells and the wild-type mast cells in terms of the amount of F-actin in the cell, and most of those differences are actually inside the cell and not at the perimeter of the cell. So we decided to determine whether the ability of these mast cells to degranulate in the absence of drin was because they can't reorganize and reduce the amount of F-actin in the cell. And so we took advantage of a compound called latrunclin B that is able to reduce F-actin superstructures in the cell. So if we treat mast cells with latrunclin B, we can reduce the amount of F-actin in the cell. We thought that this would allow these mast cells to be able to degranulate. And that's actually what we found. In this experiment, what we did is we took wild-type mast cells and treated them with increasing concentrations of an allergen. And you can see that wild-type mast cells degranulate, whereas the drin-deficient mast cells do not. But if we treat these drin-deficient mast cells with increasing concentrations of latrunclin B, we can gradually restore mast cell degranulation in these cells. So what I told you then is that this compound BTP is able to inhibit mast cell function. It does so by reducing calcium influx and therefore reduce degranulation and cytokine secretion. I told you that we were able to identify these trifluoromethyl groups at the 3 position on this compound as a really important moiety for its activity, and that if we mutate or if we change any of these residues here, we affect the activity of the compound. We use this information to purify proteins that would interact with drin, with BTP, and we found that this actin-binding protein drin is able to bind to BTP, and that these two positively charged lysines are able to interact with those negatively charged trifluoromethyl groups. And I showed you that using genetic knockout approaches, we generated mice that lacked drin and could show that in these mast cells, they have significantly disrupted F-actin dynamics, and we think that that's really important for how drin regulates mast cell function. And so I want to leave you finally with a remembrance. In a couple of weeks, we will celebrate this day when segregation of Mexican American women in California was declared unconstitutional by the Federal Appeals Court for the 9th Circuit. And so with that, I'd like to thank you for your attention. Thanks again for the ASBMB for the recognition, and I appreciate your time.